In this video, we're going to continue talking about chapter number 4, cells and organelles. So let's start. In the previous section of this lecture, we were talking about how did the first cells come to be. In other words, how life appeared from a cellular perspective, how the first cell appeared. Then we move into the properties of strategies of the cell. Here I skip many components. Why? Because you have heard about that in a lot of detail, but you can read that in your textbook if you have any doubt. Then we move into the main difference between these three domains: bacteria, archaea, and eukaryote. And the key here is that although we used to say that bacteria and archaea, they were very similar because they were prokaryotes, what I wanted to show you is the current information showing that they seem to be very independent, like three independent groups, that they have similarities and difference. And now I'm going to summarize 6 points. I'm not going to cover all of them because I'm sure that you are tired of some of those, like what is a prokaryote. But I'm just going to summarize the information to continue to the next point. So in your textbook, they summarize 6 points that I'm going to focus on in the point 2 to 4. Yeah, and I'm going to give you new information that I think is important, that and at the same time that is fascinating. So I'm sure that you remember that eukaryotic cells have a true membrane, and I don't need to talk about that. But what I found very interesting is to tell you that bacteria and archaea have our mean of storing genetic information. And when I started studying biology, I remember that many diagrams. And to simplify things in genetics, we generally, for example, this is a bacteria, so we say that it has a circular chromosome inside. And to some degree, I thought that it was like that. However, when you go and study cell biology and study in more detail, you see that it's a little bit more complicated like that. It's still circular, but it's not going to be open like that. It's going to be more like this. So it's still circular, yeah, but it's like if you have elastic band that you have to bend to some degree to to avoid tangling. So I'm going to show you that in a second. And although definitely the eukaryotes don't have the prokaryotes don't have a true nucleus. They have the genetic information is stored in a folded and compact structure that is referred as a nucleoid. So this is not happening. What is happening is that, again, we need to fold it in a slightly different way. So here you can see the nucleoid, yeah, that is the way that is the location where you find the circular chromosome that is packed in bacteria too. And here, if you want to know more about this structure, so here you can compare the difference between a nucleus that is located in bacteria versus a nucleoid. So first thing, if you read here, you will see that the nucleus is located in eukaryotes, as I said before, while the nucleoid is located in prokaryotes. Second difference is that the nucleus is a well-organized structure that is much bigger, while the nucleoid is much smaller and is poorly organized. While the eukaryotes will have a nucleus that have more than one chromosome generally, the nucleoid will have generally a single chromosome. And here you will have in the nucleus, the DNA will form a structure with histones, while the DNA in bacteria will have these NAPs that are nucleotide associated proteins. So, in other words, they will have proteins that will allow to fold the protein in the DNA in the correct way, and we will talk about that in a few slides. So again, they are not similar. The one is eukaryotes, much more complex, many more chromosomes, and in prokaryotes you will have the nucleoid that is less complex, smaller, but both need to be compact and need to have proteins that allow that the DNA doesn't tangle. Okay, and we will talk about that in a second. Again, so the membranes are very important, and to have, even for the DNA to have a complex, a lot of DNA, you will need to have a membrane, an internal membrane, and that internal membrane surround the nucleus. So now the next difference between these three domains is the presence and absence of exocytosis and endocytosis. So eukaryotes have exocytosis. Exo, yeah, it means outside. So materials, chemicals that go outside of the cell, and endocytosis is from enter. So cells, materials that need to go inside. And how they go inside is not that that bacteria and bacteria, they don't interchange any material with the environment. Is that in eukaryotes you need vesicles, yeah, so you need these membranes, these structures, very tiny structures that have lipids that allow to pack very well. uh, Here, I don't know, you can have different structures here, different chemicals here, and they need to go inside, and they travel with the help of microtubules, and they move around the cell. Okay, so why they don't have it in bacteria and in archaea? Because they are much more simple. Yeah, so eukaryotes, they are exchanging material, and they have to create these internal membranes that allow to have different compartments in the cells. In the in bacteria and archaea, as they are generally much smaller, you don't create those big compartments. Yeah, yeah, you just have all the in the cytoplasm. Most of the things are there, and they have the same pH. As I will talk later, for example, in the eukaryote cells, you have a mitochondria that will have their own pH, and that is very important for their function. So to have vesicle is a proper packing that allow these molecules to exchange. Okay, and these why vesicles will have lipids? Yeah, why they need to be packed with lipids? Because they need to, and we will talk about that. They will fuse with the membranes, with different membranes, to properly go inside and outside of the different organelles. And we will talk about that in more detail in future lectures. But the idea is that, remember, eukaryotes have exocytosis and endocytosis, while the bacteria and archaea, they interchange materials, but they don't need to pack things in vesicles. And I'm sure that you have heard about the organization of the DNA, and maybe in your introductory biology they show you that the DNA have this double helix, and that you need to pack it properly in these proteins that are called histones, that they give two turns, all this information that you can read in your textbook, and that this allows that it doesn't tangle, so you don't lose information, and they form these chromosomes that are just all these very well tangled DNA, molecules of DNA that have histones. And if you study genetics, we tell you how many turns, how many histones, how many proteins each histone it has, all this information. Okay, so I'm not going to go into a lot of detail, but you can read in your textbook. What I'm going to tell you is um, what happened in bacteria. Yeah, that for me is something new. The information that they talk about that. Okay, so we already talked about this diagram before, and here I show you that bacteria, yeah, it doesn't look like that, the chromosome. Yeah, if it was like that, when you cut it with an enzyme and it become linear, yeah, you take scissors, molecular scissors, and you cut it and you open it, it will perfectly fit in a bacterial cell. That is not true. Yeah, that is just a simplification. If you cut the DNA of a bacteria and you make it, yeah, in any of these places, it's not going to fit inside of the cell. Yeah, If you extend it, that DNA is too long. Yeah. Why? Because it's properly packed with proteins that are not histones, but they have a similar function. So let me show you about that, and I will show you a video. So I find this pretty cool. Here you can see how a bacteria and archaea pack their DNA. So what they do is they have proteins, similar to what happened in eukaryotes. Here you can see in blue all these proteins that I'm showing you here, and this is the DNA. Okay, so you're forming these loops that are twisting, all these loops. And after that, and I will show you in a video and in more slides, there is another layer of compactation that is called supercoil. So there are different ways that the circular DNA of bacteria and archaea get folded and packed tightly. Yeah, and when you remove these two layers, yeah, the twist, the loops, and the supercoil, this is what happened. So let me put it in yellow. Here you have the bacteria, and when you release the supercoiling, what you're going to end up is with a DNA that is much longer that it will not fit in the bacteria. Okay, so the DNA in bacteria and archaea need to be properly packed, and they have enzymes and proteins that are doing that. So I'm going to show you a video that explains this in a simple way, and then I will show you the summary of the video, showing you two more slices.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase I, twists are introduced in the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleoid of the bacterial cell.
Okay, so now I'm sure that you remember the two layers of compactation. So one way to compact is that from this circular chromosomal DNA, what they do is they have these DNA binding proteins that create these loops that allow to fold the chromosome 10 times. Yeah, but it need to be more compacted, and to do that, they have a second layer of compactation. So I'm just going to tell you that here there is a lot of information that you can find in your textbook. I already created the slide, but again, you already should know this. If not, you can review the slides and read the textbook about how it's specifically done in the eukaryotes. And here there is another diagram of how to go from, again, the double helix to the linear chromosomes that is what happened in eukaryotes. Okay, this is a topic that I'm also not going to talk because you have heard about this multiple times. That is the segregation of genetic information. how in bacteria and archaea you have binary fission. Well, in eukaryotes you have mitosis. That possibly you have heard about this in every single course of biology. So, if you have any question, you can read the textbook. And here I create even a slide that it can help you to summarize what is in the textbook in case that you have any question. But I'm sure that a quick read will be more than enough to remember some of these topics. And here, this is a fascinating topic that we already talked. I'm not going to cover a lot of this, but I'm sure that you remember that in eukaryotic, the transcription and translation are much more complex than in bacteria. So you have intron and exons, and the introns are not they are not involved in the final protein. Yeah, only the exons are the one that are going to be in that, and you have a process that is called RNA splicing, where you remove these green areas that are the introns. Yeah, and in the table uh, that of the previous video lecture, we talk about that these three domains have different degrees of regulation, yeah, and of this genetic expression. So just that you remember, yeah, all the things that, and if you have any doubt, you can give it a read, or you can watch again the previous video lecture. Here, I think this is, you already have talked about that before, but so I'm not going to get into a lot of detail. And you have heard about this simultaneous replication, transcription, and translation that they take place in the same in the cytoplasm, yeah, in the same location, because there is no nucleus. So I don't have to tell you anything about that. If you remember the previous video, we were talking that the archaea DNA expression is more similar to eukaryotes than bacteria. They have initiation places they share initiation places with eukaryote. That is fascinating, and that we are not going to talk a lot about that, but just that you remember again that this is an example that there is a lot of variation, and in some topics eukarya and archaea are very similar. So with this we finish the last section of the difference between these three domains. Now I hope that it's more clear what are the difference between these three domains, and in the next section we will move into the internal membranes and the importance of them to segregate functions.